In the next 45 minutes or so, I will go over the following topics in relation to the bidirectional brain-gut microbiome communication. I'll talk about how the gut communicates with the brain and how the brain communicates with the gut. I'll mention the cognitive dimension of these communications, go over some developmental considerations, and at the end present the emerging ecological view of brain-gut microbiome communication. This quote has become quite popular now that almost everybody is familiar with brain-gut microbiome interactions, with the concept. But the question of what the brain has to do, what role it plays in this communication, is less well understood. The idea that there is the microbes are able to communicate with the brain is mainly based on experiments coming from two laboratories in Canada and Ireland over the last 10 years or so that have shown that the microbes can play a role in the modulation of nociceptive reflexes, meaning pain responses, emotional behavior, social behavior, ingestive behavior, and there have also been some biological readouts associated with these changes in behavior. Should be emphasized that many of these studies are based on comparing germ-free animals with all their multiple alterations in physiology with animals that have grown up with a normal pathogen-free microbiota, and that behavioral responses, reflexive responses, are obviously not the same as complex human cognitions and emotions. Nevertheless, based on these exciting early studies, a variety of human brain-gut disorders have been postulated with involvement of the microbiome in their pathophysiology. This is fairly well established for hepatic encephalopathy, both in human and in animal studies. There's some evidence also supporting this in obesity and ingestive behavior. But then I put a lot of question marks on the other entities listed here because we do not have definitive answers if the microbes really play a role in these disorders. And that can also be said about food sensitivity. But nevertheless, I will try to discuss some of the areas or some of the evidence from clinical and basic science publications that suggest this may actually be relevant. Let me start out with the concept of bidirectional brain-gut communications. I favor the view of a systems biological view of these interactions where the circuits in the brain, referred to as the gut connectome, are intricately connected to the circuitry within the gut, including the microbes, and implying that any response at the brain level will be reflected in changes in the gut level and vice versa. So following this sort of engineering view of this concept, one can see that any brain perturbation, which occurs primarily in the form of psychosocial stress, but can also be physical stress, cold temperatures, hot temperatures, dehydration, will lead to a change in the gut connectome, and that any perturbation at the gut level, in terms of diet, antibiotics, infections, inflammation, is almost always associated with changes at the brain level, no matter if the changes are subliminal. So the majority of those interoceptive inputs in the brain are not consciously perceived, nevertheless cause reactions, functional and also structural, at the brain level. So at the gut level, the readouts, the consequences of the perturbations are in terms of GI function, secretion, motility, blood flow, and immune activation. At the brain level, the perceptual hypersensitivity to a variety of, this is not limited to food, but also to drugs. Everybody has seen the patients who get side effects even to homeopathic doses of some drugs, and also to any other visceral signals such as contractions, distensions, as is a hallmark of IBS patients. There's also other responses of the brain, such as anxiety, depression, general well-being, and so forth. When we look at it from the bottom up, so how does the gut communicate with the brain? I think it's important to first understand why there would be such a communication. Why would the gut and its microbes be able to talk to our complex nervous system? How could these primitive organisms do that? And it's a good idea to remember that the microbes have been on planet Earth for much longer than we have, living in the vastness of the Earth's oceans for more than 3 billion years. And during that time have had time to perfect their ways of communicating with each other through many signaling molecules. About 500 million years ago, the first, for the first time, it's estimated that some microbes decided to change their nomadic lifestyle in the oceans by becoming sedentary inside these floating digestive tracts of the most primitive marine animals, the hydra. And these animals are interesting because they're basically a floating GI tract with a surrounding nerve net, very reminiscent and homologous to the enteric nervous system. And these animals have been able to generate peristaltic reflexes and changes in the direction and effectiveness of these reflexes depending on the different conditions. So what happened once the microbes settled inside these in these animals through lateral gene transfer, many of the genes that were responsible for producing neurotransmitters were exchanged with this primitive nervous system, made it became neurotransmitters in the enteric nervous system, and later when animals developed a polar architecture, were transferred into the central nervous system. So the fact that today we can identify almost identical molecules that communicate, that allow communication between nerves at the enteric nervous system and central nervous system level originally really came from the gut microbes. So the microbes clearly have the ability to speak the same language and to communicate with our brains. This is just an example of that many of the enzymes necessary to convert amino acids and other nutrients into neurotransmitters are present in many of the microbes, which are able to produce a large number of neuroactive substances from the undigestible food that we ingest. There are several communication channels between the gut microbiome and the brain. I like to demonstrate this here. If you go from the left side to the right side, so short-chain fatty acids can get into the systemic circulation and possibly reach various targets in the body, including the brain. Immune cells and their signaling molecules, cytokines and lipopolysaccharides, again can move through the gut epithelium and get into the systemic circulation. But also, as shown here with the light blue line connecting them to the vagus nerve, they can signal in a neurocrine fashion through the vagus nerve. Many cytokine receptors have been identified on afferent nerve terminals. Then moving further to the right, a series of cells called enteroendocrine and enterochromaffin cells contain either neurotransmitters or gut hormones that can be released into the systemic circulation, but similar to the inflammatory mediators, can also activate vagal afferent terminals and signal to the central nervous system by that mechanism. And then there's, as we just mentioned, several neuroactive substances, neurotransmitters, which again can either go via the systemic circulation to the central nervous system, but certainly also can modulate the circuits of the enteric nervous system, influencing peristalsis, secretion, and other enteric nervous system outputs. First, focus on the one particular cell that has been studied in great detail, the enterochromaffin cell that contains more than 95% of the organism's serotonin. So these serotonin warehouses are intimately connected not only with the brain but also with the microbes, and this is illustrated here. It's information that really has just become known in the last couple of years. Two pieces are particularly interesting. So one is that these enterochromaffin cells have these nerve-like extensions, these so-called neuropods, which then form synapse-like connections with afferent nerve terminals, most likely the vagus nerve, shown in red here, that carry that signal, can carry the signal into the brain. Equally interesting is the fact that these enterochromaffin cells not only have receptors for many of the signaling molecules that the microbes can produce, such as short-chain fatty acids and secondary bile acids and others, which can activate the cells to release serotonin, but also, as has recently been shown by Elaine Zhao at UCLA, that a significant portion, probably the majority of synthetic activities or serotonin synthesis in these cells by activation of the tryptophan hydroxylase is a consequence of stimulation by metabolites produced by the microbes. If we take food into the account, which obviously influences the behavior and gene expression and metabolite production of the microbes, we have a continuous system of events or chain of events from the microbes stimulating the synthesis and release of serotonin onto vagal afferent terminals that carry that signal directly into the brain. So in addition, just emphasizing this concept of the gut connectome, there's also bidirectional interactions between immune cells in the gut and these enterochromaffin cells. The vagal connection has been implicated in many animal studies that have shown effects where gut microbes were manipulated and caused behavioral changes. And these vagal pathways, we can speculate they also play a role in the observed effects in humans. One of the very few studies, probably the only study so far that has definitively shown this, where perturbation of the gut microbiome can cause changes in the activity of brain networks associated with emotion recognition. This is illustrated here. In a study in healthy young women, Kirsten Tillich within our group showed that the daily ingestion of a commercially available probiotic mix in form of a yogurt was able to produce changes in the brain, which were tested with a emotion recognition task, where individuals had to match the emotional face on the top with the same emotional expression of the two lower faces. The brain networks that are engaged during this task are shown on the right in these fMRI images. Without getting into the details of the individual brain regions, let me say that the main difference between the placebo group, actually were two placebo groups, and the active treatment group, was that the connectivity between these brain regions decreased with the probiotic with the probiotic ingestion. Even though there were no subjective changes in mood and affect in these healthy individuals, one can assume that this would lead to a decreased emotional reactivity to emotional situations in the environment. We don't know what the mechanisms underlying these changes, because obviously we can't do a vagotomy in these individuals. But we know from a previous study that with the identical interventions, that the metabolites that the gut microbes produce are altered during this 4-week intervention, whereas the composition of the microbes did not change. So I show this slide just to emphasize that we do have some evidence that these mechanisms identified in animals do apply to humans. On the other side, to emphasize that there are very few studies so far that have been able to demonstrate. Another mechanism of by which the gut microbes can signal to the brain, important mechanism, has to do with the immune system. This concept that changes in the gut microbiota can affect, can induce a low-grade immune activation, which then either through inflammatory mediators in the circulation, or again through activation of vagal afferents, which can encode these immune mediators and can signal to the brain. And let me show you one example that that has been supported by a substantial number of experiments in animals, how diet-induced microbiome perturbations can result in local and systemic immune activation. So the main intervention has been a high-fat diet given to mice. So let me start out with the top panel on this. On the left side, you see the normal interactions between the gut microbes, a healthy gut microbiome, and the gut wall, both the mucus layer and the epithelium. On the right side, you see the changes that have been observed in mice on a very high fat diet, 60% of caloric intake in the form of lard, and a reduction or elimination of undigestible carbohydrates. Two things happen under these conditions. One is that there is a reduction, significant reduction of the mucus thickness, and there is an increase in the intercellular permeability. These mechanisms together resulting in a reduction of the gut barrier with increased access to the gut immune system and the systemic circulation of substances, inflammatory substances such as lipopolysaccharides, but also cytokines. And this mechanism, referred to as metabolic endotoxemia, has been implicated in diseases such as obesity, diabetes, and metabolic syndrome. It has been speculated that related to similar immune activation can play a role in the neuroplastic changes in the brain of patients with depression, anxiety, and one can speculate that they may even play a role in food sensitivities. So this metabolic endotoxemia can affect adipocytes, leading to these inflammatory reactions in fat cells, but also the brain. Just to illustrate the process again, something that we know from studies in animals, that inflammatory mediators generated at the gut epithelial interface ultimately result in the generation of cytokines, some of which by different mechanisms can cross the blood-brain barrier activate the microglial cells. Keep in mind that there's at least as many microglial cells in the brain than neurons, but then the activation of the microglial cells resulting in cytokine production and so-called neuroinflammation. This neuroinflammation has been implicated in, for example, in the decreased, in the inflammatory changes in the hypothalamus, underlying development of resistance to satiety hormones, as well as leptin. They have been implicated in neurodegenerative diseases, chronic pain, and chronic inflammation. We are currently studying these mechanisms in patients with inflammatory bowel disease, which show patients would show neuroplastic changes, most likely as a consequence of this neuroinflammatory process. Now that I've shown you some of the mechanisms by which the gut can communicate with the brain, let me present some equally interesting data and mechanisms that show how the brain can also communicate quite well with gut. So, as I mentioned earlier, brain perturbation from psychosocial stress, physical stressors, can engage output systems in the brain, the autonomic nervous system and the HPA axis. And the mechanisms that play a role in this activation of brain outflow systems is the way that individuals perceive the stressor, also the way they develop negative predictions. So, for example, this concept of catastrophizing, assuming a high likelihood of worst outcomes, and so related to this, the expectation of a negative outcome, which is a hallmark of the nocebo response. The gut responds to this influence by the brain in terms of altered secretion, motility, visceral sensitivity, blood flow, and immune activation. And let me give you a couple of examples from two different areas of not food hypersensitivity, but of autoimmune disorders. So one in terms of asthma, where it's been shown that the interaction of stress and food allergy is involved in the flares of patients with asthma. And the investigators showed that it was the perceived stress that predicted the flares, shown on this graph at the bottom. On the x-axis, the perceived stress ratings, and on the y-axis, the disease activity. A different autoimmune disease, inflammatory bowel disease, shown by the group of Charles Bernstein at the University of Manitoba, who showed that a relationship between perceived stress, symptoms, and inflammation in patients with both UC and Crohn's disease. In summary, in this fairly large sample, the investigators showed that perceived stress was associated with increased symptom report for both patient groups, but surprisingly that there was no significant association between perceived stress and intestinal inflammation for either disease group. They concluded that also there was a strong relationship between perceived stress and GI symptoms. The perceived stress was apparently unrelated to concurrent intestinal inflammation, at least as was indexed by fecal calprotectin levels. And we'll come back to this, how the brain may generate increased symptoms even in the absence of a gut-related or immune-related component. But before I do this, let me just show you there's clearly important influences of the brain on the gut-based immune system and its activation. I will not get into the details of the players in this, which have been, I assume, already discussed extensively by other players who are much more expert than I'm in this area. But just showing so the schematic of how the normal gut is altered in the case of food allergy, with an increase in or with a decrease in the barrier function, increased access of allergenic components of food to the gut immune, to various cells, the gut immune system, and the activation of mast cells. So the reason I'm showing this, because like the great majority of these review articles and schematics shown essentially leave out one important modulator of these systems, both in health and in disease, namely the central nervous system. And let me show this on the next schematic. It illustrates the various cells and mechanisms that have clearly been shown in experiments that are susceptible to influences from the brain in form of the autonomic nervous system, which is engaged obviously in response to stress, and changes are associated with various emotional states such as anxiety, fear, and anger. So let me start out with the mast cells, left lower corner, which have been shown to they can be degranulated and activated by the sympathetic nervous system, been shown many years ago by John Bienenstock with conditioned fear responses. So even in the absence of an allergen, that a conditioned response could activate these mast cells, leading to consequences similar to allergic reactions. If you go up from there, you can also see that the sympathetic nervous system can clearly increase the gut permeability, change the interepithelial permeability, also change mucus production, and probably the layer, the thickness of the mucosal component of barrier function, and also change pH function. Then, if we go in the middle of the slide, as I mentioned earlier, the autonomic nervous system can activate not just the enterochromaffin cells, but probably most of the enteroendocrine cells in the gut to release their contents into the gut lumen, affecting microbial behavior. And finally, experimental evidence that the sympathetic nervous system, the vagal system, and the HPA axis all can influence the activity and function of macrophages. Just to illustrate this here schematically, how glucocorticoids, as a consequence of HPA axis activation, vagal efferents, and sympathetic postganglionics all can modify behavior and responsiveness of macrophages to microbial products. Here it's shown really the output of the brain in response to stress with the activation of the autonomic nervous system by this chemical switch in the brain, corticotropin releasing factor. And finally, and certainly not the least interesting of these brain influences on the gut shown here. So we all know that norepinephrine increases in stressful situations, fear response, anxiety, and that the main transmitter of these responses, sympathetic nervous system responses, is norepinephrine. We now also know that norepinephrine is secreted into the lumen of the gut, where it can interact with adrenergic-like receptors on microorganisms, changing their gene expression pattern, increasing their virulence, and making them produce norepinephrine-like substances, which can then in turn interact, feedback on adrenergic receptors on the gut epithelium. Now, for a large number, this modulation of growth and gene expression patterns for a large number of microorganisms, both pathogens, mainly pathogens, but also some commensals. Even though not relevant really to this discussion, this includes H. pylori, and it's intriguing to speculate that this long-held or the extensive evidence for a role of chronic stress in ulcer pathophysiology may be related to the changes in H. pylori behavior mediated by a norepinephrine's change in its gene expression pattern. So this discussion about the brain-gut microbiome axis in food sensitivities would not be complete without mentioning the cognitive dimension. What's illustrated at the bottom is a simplistic version of, or a simplified version of complex neurobiological mechanisms that have been identified underlying the placebo response. There's an extensive literature now that specific brain circuits resulting in outputs to the GI tract and other areas of the immune system or the body are engaged during the placebo response. Less studied, but certainly supported by indirectly by many studies, is that different types of mechanisms are mediating the nocebo response. An important factor here is that we have memories encoded by learning of previous situations, which affect our the assessment of salience by the brain and the generation of expectations of what our brain thinks will happen based on previous experiences. So this is important because our patients and the general population are bombarded with semi-scientific information by fields like the functional food, the functional food community, and several best-selling authors that have really started this I don't want to call it hysteria, but change in or attitude of people towards food in general and towards some specific things such as gluten and the detrimental effects of grains, or all kinds of grains on our nervous system and our brain-gut axis. So a few New York Times bestsellers have created a semi-religious movement against all grains, which has converted millions of people worldwide. I've heard these stories from individuals in many different countries around the world to follow the grain-brain diet and blame a wide range of symptoms that encompass everything from fibromyalgia to depression to chronic fatigue and IBS on the evil effects of grains, and also a parallel message on the benefits of high fat in our diet. I strongly believe that the pandemic of gluten sensitivity, the non-celiac gluten sensitivity that has occurred around the world in the last few years, almost coinciding with the publication of the Grain Brain book and more recently the Brain Maker, is largely due to some kind of a variance of the nocebo response, where people expect the negative effects of certain food components, now particularly gluten, wheat components, that would trigger symptoms. If these symptoms are actually reflected in changes at the gut level, which up to now have not been identified, or if they're simply generated within the brain in terms of symptom generation, needs to be determined. Developmental considerations are important in understanding the possible role of brain-gut microbiome interactions in food sensitivities. There is now rapidly growing evidence that possibly the most important phase in the programming of the brain-gut microbiome interactions occurs early in life, starting prenatally during pregnancy, and continuing really to the age of 18 to into the early 20s, when brain development is completed. A very important phase, the first 3 months of life, where the gut microbial architecture is established, where a lot of signaling occurs from these emerging gut microbial communities to the brain, influencing really synaptogenesis and development of these brain circuits that ultimately lead to the adult brain networks that generate the outputs of the brain. And if this development is altered early on in life, form the basis for many brain disorders. I'm convinced that certain gut microbial metabolites, possibly also influenced by viruses, play a very important role in these early shaping and programming of the gut-brain microbiome interactions. And we know from many studies that the factors that play such and contribute to this programming are prenatal. We know that stress, maternal infections, maternal diet all play a major role in influencing both the development of the gut microbiome, but also brain development. Then at birth, factors such as early adverse life events and antibiotic consumption, I'll come back to this in a minute, have a major influence. And finally, that the mode of delivery and the early nutrition for the gut microbes also play major roles. So this may be the period in our life that has the key to the understanding of how microbes are altered and how these alterations can influence brain function. Let me come back to the role of probiotics. This has been emphasized and mentioned in great detail in Marty Blazer's book, Missing Microbes. So these are just the numbers that hit home the message of how extensive this influence of early life antibiotics are on the third bullet. So 2.7 courses of antibiotics by the age of 2 and 11 courses by the age of 10 on the average that our infants and newborns are exposed to, with detrimental effects on not just on the microbes and their diversity, but also as a major risk factor for metabolic diseases, obesity, and in my opinion also for many of the other brain-gut microbiome disorders that manifest later in life. My preferred view of the brain-gut microbiome interactions is an ecological one, as been proposed by many other people, including David Relman. An understanding of the gut microbial landscape in terms of a stability landscape with valleys and mountains, with the stable states being the valleys, perturbations leading to switching or to transitions between different stable states, and with influences such as early life experiences, genetics, and other environmental influences on the shaping of that landscape. So once the landscape is shaped early on in life, it's fairly stable, but can still be affected by other factors, in particularly diet and drugs. So we talked about the high-fat diet, which can change the interactions of, which can influence brain structure and function and responsiveness, but also by various factors related to our brain, such as stress, particularly chronic stress, cognitions, as we talked about in terms of these nocebo responses, and also other components such as social connectedness. So rather than looking at it as a linear system by which the brain, the gut, and the microbes interact with each other, I think there's two views that are kind of complementary. One is the systems biological view with which I started my presentation, and the ecological view, both of which are consistent with the model that I've laid out here. Not a single factor can explain all the changes, but multiple factors make contributions to the overall output of this system. So in summary, the gut microbiome axis plays an important role in food sensitivities. I think, I hope I convinced you during my presentation that there's evidence for multiple mechanisms involved in this that underlie this role, such as the bottom-up and top-down influences that we discussed, that early life factors play a major role in the programming, not just of the gut microbiome, but also the entire axis, including the brain. That stress can alter the gut connectome, including barrier function, immune function, and gut microbial behaviors. I think a very important addition to the traditional gut immunology concepts that do not take the brain as a major player. And including the gut-brain microbiome axis in any pathophysiological model is consistent with the epidemiological studies about the role of diet, perceived stress, and antibiotics in symptoms, food hypersensitivities. And then finally, important for less so for the food allergies, where this probably does not play a very prominent role, but certainly in non-allergic food sensitivities, the cognitive factors may play an important role in some of these disorders. If you like to learn more about the brain-gut microbiome interactions and the mind-gut connection, please don't hesitate to buy my book, which has just come out, which gives many examples and including the topic of food hypersensitivities. Thank you for your attention.